where we said we would like to have insight from most different kinds of fields into the future of this personalized medicine. And the first to speak to you about what personalized medicine could be in the implication, and this year we had really a lot of regulatory authority talk, but I think it is really great that you, Frank Weichold, stay and will tell us now what your vision is about this.
Hi everyone, thank you Beat for organizing a wonderful meeting which is very unique I think in the world and has a very high appeal to me personally, but I also think it's a forum to discuss questions in a quite different way than they are usually discussed. Now I have to admit, Beat, I did not make a philosophical talk this time because I wanted to use the opportunity to expose the audience and those who follow up with the information to somewhat the current situation about personal medicine and how the regulator, specifically FDA, is approaching it. And as such, I will share some information with you as to the history of the past decade perhaps, and that then go into some of the challenges that we are facing up to. So, you know, defining things as personalized medicine is sometimes a question of interpretation. I think in some way human beings understand what it could be. Different organizations give different definitions. Some of them drive it with a specific purpose in mind, and as such, I think you can debate what is the most useful information. I think the Personalized Medicine Coalition restricts it somewhat. They don't necessarily include the aspect of social and behavioral science, which in my mind is an important element to it. You see the European Union, right treatment, right person, right dose, right time, straightforward. And, you know, President's Council of the Advisor on Science, the tailoring of medical treatment to individual characteristics of each patient, I think it meets the bill. And then we have the Institute, the NIH, which states that it is a form of medicine that is informed by each person's unique clinical, genetic, protein and environment to prevent, diagnose and treat. I think it's rather complex. I may have phrased it a little bit different, but I think you get the idea. So as such, that's quite useful. Now, we're all familiar with the fact that no single treatment fits perfectly all individuals. And this is just to reiterate the fact that even in simple questions like dosing, you have different response rates and different responders, which is encoded by the genetic makeup. But in that context, I also would say the simple fact that we have to understand more and better what is going on here will have significant implications towards our approach to develop new treatments and drugs, because if we do not manage to reduce the time that it takes from making a decision to put something into clinical development towards getting a licensure to put it on the market, I think the industry and the healthcare systems and the society will face significant problems. So I mentioned that yesterday in some of the discussions already, that it is quite important to shift the risk from the actual clinical development before approval into the phases after approval, and we need to make this a part of the evaluation and assessment period. What that requires, however, is a lot of investment into, let me call it, logistics and tools that give us a very precise and nimble feedback about the well-being and perhaps the disease of an individual. So how can a regulator get involved, and what are some of the responsibilities? Well, we have to be quite careful. We have to evaluate risks versus benefits. That's nothing new in the context of personalized medicine, because that is always the big question. We certainly have to keep up with science, and in some ways I believe the regulators responsibility is to identify critical issues and drive the science because of the unique position that the regulator has. It is also important that the regulator strives to provide clarity, predictability, and guidance to industry and not build hurdles that will discourage investment into one or the other direction. We want to make sure that the scientific information that is being generated is appropriately used in clinical practice, but I also want to point out that FDA does not regulate clinical practice. In a sense, it doesn't take influence into how physicians apply the medicines. It's a question of collaborative work among the stakeholders, and the aspect of diagnostics is very critical, and it was discussed today as well as in the past sessions. So we are interested in streamlining the regulatory process, obviously, and advancing the science as a tool to help drive innovation, and that is part of personalized medicine as well. Now let me give you quickly a historic perspective on it. I think at least for the past 10, 15 years or so, the recognition of the individualization of medicine is reflected by certain activities. The National Center for Toxicology Research established a Center for Excellence in Bioinformatics, Functional Genomics, and Structural Genomics. The Center for Devices and Radiation Health creates an office for in vitro diagnostics and device evaluation and safety. CDER launches a voluntary genomic data submission program, and many of you are familiar with it. Only 2 years later, we have CDER that generates a genomic and target therapy group in the Office of Clinical Pharmacology. CDRH recognizes or reorganizes the office in science and technologies, and in 2006, National Center for Toxicology Research again reorganizes the division of pharmacogenomics and molecular epidemiology into a new division of personalized nutrition and medicine. 2009, CDRH creates a personalized medicine staff within OIVD. The Commissioner, Dr. Hamburg, launches the FDA's Advancing Regulatory Science Initiative, something I mentioned before, and defines some of the priorities there. The Center for Biologics Evaluation and Research develops a consortium of intramural research focusing on the aspects of personalized medicine. 2010, CBER again launches the Genomics Evaluation Team for Safety. In 2011, Commissioner Hamburg restructures FDA, creating 4 directorates with the idea in mind that common and synergistic activities can be coordinated by the directorates. In my mind, the unfortunate aspect of this is, in theory, that's a good idea, but one also has to assign resources to a directorate. Otherwise, it's very difficult to coordinate things, and that obviously was not implemented. The strategic plan for regulatory science and the reflection of personalized medicine, specifically in the second priority in this plan, was also established in 2011. And we have Intercenter Drug Diagnostic Collaborative, which is established with representatives from CBER, CDER, CDRH, and OMPT. CBER creates a personalized medicine team. 2012, there are some reorganizations towards addressing personalized medicine, specifically in biomarkers and alternative models, innovative safety and toxicology, and personalized medicine. And in 2013, the FDA launches the FDA Genomic Working Group. So that information is just for you to think about the agency and what has been done there, but also to recognize perhaps gaps of things that have not been addressed. So, defining regulatory pathways for medicine. The development of regulatory review for personal medicine products raises challenges. There are not just regulatory origin, there are policy, there are coordination of sponsor activities, and certainly there is review management involved. The success of personalized medicine depends on a safe and effective diagnostic. Again, that's very much part of developing a personalized medicine strategy. There are, however, questions that remain about the application of evidence standards and the regulatory oversight of diagnostic tests and its approvals. Personalized medicine involves the use of 2 or more medical products most often, and as such it is just like in real life a quite complex aspect to tackle. So thinking about the regulatory pathways that are to be considered in the context of personalized medicine, CDRH and CBER, as well as the Office of Special Medical Products, are responsible for establishing regulatory pathways and policies that fit the bill for personalized medicine. So there is an initiative and working group, and there is an assigned responsibility within the agency. The policies generated and provided will form guidance on the incorporating of biomarkers in drug development processes, the designation and the designing of clinical trials in incorporating those biomarkers, as well as the evaluation of pharmacogenomic data. There is, of course, always the question of the companion diagnostic tests and how to use them and how to qualify them. So those are challenges in the context of pushing progress in personalized medicine. Now, since we have a meeting on nanotechnology, nanotechnology and development of in vitro diagnostics, we face challenges. For example, in the context of the whole genome sequencing, we had wonderful presentations during the meeting, so I don't have to reiterate most of it, although I would like to point out that at the current time we are still struggling with the precision of the sequencing processes, and as such that's something that we have to keep an eye on as a regulator, and certainly we need to work with the stakeholders together to understand how to make this a very reliable, reproducible and precise method. The whole gene sequencing could have significant clinical and public health applications and implications, obviously. So some examples of the whole genome sequencing for regulatory implications. Identifying genetic variations associated with the treatment of adverse events is an initiative that is mainly handled by the Center of Drugs Evaluation and Research. The evaluation of development of resistant variants after treatment, again, is a CEDAR initiative. Detection of infectious agents to evaluate blood, vaccine, tissue, and cell therapies, as you could imagine, is an activity in the Center for Biologics Evaluation and Research. And CDRH focuses on the evaluation of new diagnostics. To assess the technical performance and clinical validation of whole genome sequencing is one of the responsibilities of the only center that we have that's not involved directly in regulatory processes, namely it's the National Center for Toxicology Research in Arkansas. Detection and prevention of blood-borne pathogens is obviously a very important element that plays a role, and if you think of the statement that we are what we eat, I think that creates a context into personal medicine. But there are also challenges in regulation of sequencing methodologies and processes. We currently do not know how the significance of genetic variations is to be used and how we apply that into the clinical process. If we think of rare events and if you think of therapies in this context, it is very difficult to validate some of the information, particularly if a patient group is very small. Demonstrating the validity of each variance may not be practical or even feasible. It's also a question how to integrate genomic information in the clinical practice while ensuring the safety and efficacy of genomic tests. So if you want to make clinical decisions on the basis of a sequencing testing, I think you have to have some confidence, and certainly a process of validation has to be taken place. And again, that's an opportunity for standardization and harmonization. As you can imagine, with big data sets, you have IT challenges, and I can share with you that the agency has made a lot of progress in that, also in the context of putting information in, using the cloud as a medium, but not all the problems are solved. And I think it's a question of standardization and applying new technology, and also understanding the life cycle of IT resources, which sometimes can be a challenge in investing into it. So FDA has policies to develop new methods and to evaluate whole genome sequencing tests. I think one of the important aspects is to establish the analytical validity, but it's just as important to understand the clinical validity. And the FDA believes that the clarification around the regulatory oversight of the framework for in vitro diagnostics would facilitate the development of in vitro diagnostic tests for use in providing optimized treatments for patients. Nanomaterials are often regulated as combination products, and we've discussed the challenges in combination products and who takes the lead. For example, when you talk about 7 centers that essentially are specialized and focused on a particular entity or a particular commodity. But there's also a challenge in the context of not just a particular product. If you have a diagnostic test that relates to the therapy, then I think the case just becomes even more challenging. So, as I shared with you, FDA tried to address that by specifically having created an Office of Combination Products, and as such, some of the challenges are there at least coordinated through one resource. The question of co-developing regulatory policies and challenges is eminent. It is essential for the development for personal medicine. Combination products are regulated under different regulatory authorities, for example, drugs versus devices, regulated by different FDA centers, CDRH, CDER, or CDRH and CBER, and the ownership and manufacturing is sometimes also performed by different companies. And certainly as a regulator we have to deal with that. There are considerable challenges. They are technical in nature, they are organizational, and they're also procedural. The specific challenges related to nanomaterials under the conditions is uncertain, and certainly if we deal with unknowns, there's always a potential of hazard. But nevertheless, applying the basic principles of making products safe and evaluating its effectiveness carefully, I think we do not necessarily change the principles for product approval. So, how do we overcome some of the challenges in co-development process? FDA has developed a process for helping shepherd co-development programs through regulatory channels. Elements that we have already discussed is early communication with the sponsors to ensure that the understanding about the specific issues are in a dialogue. There is an opportunity to clarify things. The opportunity to have frequent consultations both internally and externally throughout the process is certainly an important element as well. And then we have to make sure that we as an agency have a close coordination among the relevant product centers. And that's sometimes a challenge because historically the centers have developed in a relative siloed way. The FDA has published a guidance document for industry that is under the title Co-Development of 2 or More New Investigational Drugs for the Use in Combination, and it was released actually past year in June. So, when thinking of developing regulatory standards, research methods and tools in support of personalized medicines, we, as you're all familiar, we have a biomarker qualification program. This provides a framework for scientific development and regulatory acceptance for drug development, facilitates the integration and qualified biomarkers in regulatory review process, and it encourages the identification of new biomarkers. We also do have a genomic reference library for evaluating and qualifying whole genome sequencing platforms. And I think it's a critical function of the regulator to provide standards and define how such new technology can be used. In continuation of developing standards and tools, we have developed a virtual environment for computing, which we call the Hive. We have information on the web for those who are the technical gurus and have an interest how this actually works. I'm not good enough to explain even the very basic principles of it, but essentially it uses the computing power of different computers in a network towards the analysis of big data. And we also use new methodological approaches to the studying of medical device performance in the context of clinical outcomes. Now, all of that is also supported by a program that is run through the office of, or from the office of the chief scientist. And as you probably remember, I mentioned the program of advancing regulatory science in support of personalized medicine. And as such, we have some priorities defined. You can review that in the documents that are displayed here. And this is all online. I believe that the FDA plays a critical role in bringing the stakeholders together. We have to think about reforms and adaptations to the processes that are driven by science and the technological landscape. And we have to take on the challenge to modernize and streamline regulatory pathways as well. So what are some of the pillars of implementation? Certainly it requires leadership, coordination, strategic planning, and transparency. We have developed a program that supports scientific excellence. I mentioned that the centers of excellence with academic institutions, but it's also critical that we provide information that is teaching material and material that is used for the training of reviewers. So I think the transparency in the area of education and the learning processes are important to build on. And certainly the mission-critical research only can be performed in collaboration. One of the challenges for the agency, as you can imagine, if you know about the salary structure in the government, is recruitment and retention of outstanding scientists. And there is no easy solution to that as far as I can tell you. So, as you can reflect, there's a lot of activity going on within the context of science activities. This is just a list. We are strongly building on working groups, consortia, and committees. And as such, I'm not going to go through the numbers, but it gives you a snapshot. One of the specific things that I want to mention again is the broad agency announcement as one of the contracting mechanisms that's relatively nimble and often used for not only doing horizon scanning for new solutions that can be brought to the agency, but also it is a mechanism where grassroots problems that occur in the regulatory processes can be in a relatively short time period addressed through engagement with contractors that have the capacity to provide solutions. We currently have 4 centers of excellence. I just want to mention that in the academic area. This is Georgetown University, University of Maryland, Johns Hopkins University, and UCSF Stanford in a collaborative center. I think we've made some progress, but there are certainly lots of challenges, and I think if there is some time at the end of the presentations we can discuss and I can address some of your questions. With that, I'm going to make reference to a document that we recently released that is called Paving the Way for Personalized Medicine. You can find it online and you can flip through it, and I think in some ways it provides not only a historic review but also some outlook into the future, how the agency sees the challenges and how we want to address that. And with that, I'm going to end and thank you and give the word back to my colleague.
So there's time for several questions in this talk. Who would like to be first?
Yes. In general, what is the average time frame for approval of state-of-the-art drugs? Do you have that?
It's about 340 days. With that, actually, FDA is world champion.
So thank you very much for the very interesting talk. And of course, for all of us, this personalized medicine is a very important goal. And we need biomarkers in order to have a medicine personalized. So you mentioned that you need analytical and clinical validation for a biomarker. And you have a program, an biomarker qualification program. So what is the amount of data that we need to qualify a biomarker? So this, if you find a correlation, this is enough, an analytical and a clinical correlation, enough to get a significant value, or you are, how do you determine what kind of data and how much data do we need?
It's a very important question because that's the making and the breaking, obviously, to qualify or validate the not just the validation of the analytical method or whatever method it is, but to validate it in a clinical context. And that, unfortunately, there is no simple or just one answer to it. It really depends on, first of all, the disease and patient population that you look at, and certainly also the readout that you get from your method. It is, in my mind, a dialogue about the statistical approach that you take and the practicality of obtaining the data. If you deal with a very small disease group or a very unique situation, you cannot collect thousands and thousands of sample points from patients. Right, so this has to be seen in context, and I wish I would have an answer that fits all, but unfortunately there isn't. So there's truly a need to evaluate it case by case. So
just one more comment. This is also unclear what is FDA regulated and what is in-house developed tests. So I think that in this in vitro medical device field, what is under CLIA and what is under FDA is completely unclear to most of us.
I think you're right. There needs to be some work done to define and better define it. And also I think there are some things where the borders between offices have to be made less interruptive for the process. And I at least have some awareness of that, and some of my colleagues do. So I think there will be a lot of work and some changes that you will observe in the near future.
So now 2 quick questions. The first is here.
So for clinical trials done in very small groups of patients, do you see that those would fit under the current orphan drug program? Or do you envision developing a new program specifically to deal with very small patient groups for personalized medicine applications that may not necessarily fit into the orphan drug program?
I do not know that, but if it can fit in the orphan drug program, then of course we would prefer to not develop a new one, but there may be a practical reason why a new program should be considered, but I think that's something we have to also be confronted with the facts and then make decisions about.
Here? In your biomarker qualification, how important is the accuracy of the diagnostic test? Because if you really stratify patients based on a diagnostic test and it is not accurate, you may create the wrong care.
You bring up a very important point. I guess if you base your clinical validation on a relatively inaccurate test, of course, your statistics are not going to work out. So there has to be a consideration for both, but we all know that we have to deal with reality. So some of the tests, unfortunately, coming back to sequencing, there's still challenge in the actual sequencing methods and the accuracy. If you use 3 different methods, you get 3 different variations in the sequence, and it's just a simple fact. So those are things we have to take into consideration and also understanding the limitations of it.
I think we've ended the questions in this portion, but there's a period at the end of all of the speakers when there's a lot of time for questions. So we'll come back to your question then. And I think in our partnership, it's now Beat. You're going to...
We will have a second change. We will ask Rick